Students, in this class we will study about the experiments on photosynthesis. That is, in this part we are going to study the different factors which are required for photosynthesis, which will make it more clear. In order to perform the experiment on photosynthesis, we will have to follow certain steps. Like, suppose we are going to perform an experiment, then what are the steps taken for it? As a first step, we keep the plant in darkness for 2 to 3 days to de-starch the leaves. Means the first step is de-starching. That is, the leaves are made free from starch, by keeping plant in darkness for 2 to 3 days. Actually, in this process, when the plant is kept in darkness, then there is no fresh photosynthesis, and starch from the leaf is converted to glucose and translocated to other storage parts and reconverted into starch. Hence, the leaf becomes free from starch. It does not give positive taste for it. Then, the plant, when the leaf becomes free from starch, is kept in sunlight. Plant is kept in sunlight under experimental condition. Under experimental condition, that is, it means that the experimental setup is arranged according to the factor which has to be tested. That is, there are many factors like sunlight is essential for photosynthesis, chlorophyll is essential, carbon dioxide is essential, and so on. So, the plant after de-starching is kept in sunlight under experimental setup. As a third step, after few hours, the leaf is plucked and tested for starch as follows. Means actually this was the beginning, or that is a step taken for the starch test, but now we have to perform the starch test. Sometimes the question is asked that write steps required for a starch test, then the points related given in this part, studied in this part, will be written. So, in the first step of a starch test, the leaf is boiled in water to kill the cells and fix the starch. On boiling, the cells are killed, enzymes are destroyed, and the metabolic activity will stop. Hence, the starch will not be converted into another form. It will be fixed. So, in a first step, the leaf is boiled in water for a minute to kill the cells and fix the starch. Now, then as a second step, this leaf is then boiled in methylated spirit or methyl alcohol, which dissolves chlorophyll and makes the leaf free from it, such that the leaf becomes light brown or colorless, such that on adding iodine, the color change will be easily visible. So, as a second step of a starch test, the leaf is boiled in methylated spirit. methylated spirit or methyl alcohol to dissolve chlorophyll. To dissolve chlorophyll. Methylated spirit acts as the solvent of chlorophyll. The leaf becomes colorless or light brown in color, such that when iodine will be added later on, then the definite color change or clear color change will be observed. Now, in third part, the leaf is rinsed with water, rinsed with warm water to remove excess of alcohol, and make the leaf soft, which has been turned crispy on boiling in alcohol. Now, here we see that the diagram for this step, the setup of this step is taken like this. In a beaker, we take water. In the test tube, we take methylated spirit, in which the leaf under the experiment is placed, and then it is boiled. Here there is water. This is methylated spirit. Now, the question may be asked that why is this methylated spirit not boiled directly, or leaf is not directly boiled in methylated spirit? Because the methylated spirit or alcohol is volatile in nature and highly inflammable. So when it will be heated directly, then its vapor will catch fire. So this is the diagram for it. Now then, as a fourth step, leaf is sprayed in a petri dish. Petri dish actually is the circular disc made up of glass, petri dish, and iodine solution is added to it. Now, again you see, what will be the observation? The part of the leaf having chlorophyll starch, the part of leaf having a starch turns blue black, and without a starch turns brown. So this was the step of a starch test. So whatever may be the experiment or factor to be studied, these were the different steps that will be followed. For every part, this step in sequence will be taken. So now let us start with the factors to be studied. First experiment, experiment 1. To show that sunlight is essential for photosynthesis. Now you see, students, again that the steps will be taken in a similar way. That is, we will take a well-watered potted plant as a first step. It will be kept in darkness for 2 to 3 days, and then we will arrange the setup. How is the setup arranged before keeping in sunlight? One of the broad leaf is covered with a stripe of, partly covered with a stripe of black paper from both surfaces. This is black paper. Actually, black paper does not allow sunlight to diffuse in. It is fixed with the help of glass slide and clamp, and also then supported with the help of a stand, otherwise the leaf will be plucked. So this is the experimental setup. Now the setup is kept in sunlight for few hours. And after few hours, the leaf will be plucked and tested for a starch. When the question is asked, the diagram is given. The first question will be asked, what is the aim of the experiment? Like, first of all, let us explain it. So, the setup is arranged. After few hours, the leaf will be tested for a starch, and we will observe that after a starch test, what will be the observation? We see that the part of the leaf which was covered turns brown, whereas the uncovered part turns blue-black, presence of a starch, whereas the part which was covered turns brown, showing that there is absence of a starch. So, thus we see that the part of the leaf which received sunlight only performed photosynthesis and has a starch. Now, in this part you see the part of the leaf which was covered with black paper acts as control. Control means for the comparison. So, covered part of leaf acts as control. Now again, student starts. Let us start. What are the questions asked? In plant physiology, diagram-based question is always asked, where diagram will be given, and what will be the question. Questions will arise similar to that. The setup will be given. Question. First, what is the aim of experiment? What is the aim of experiment? Second, define the process. Define the process. It may be part of the question. Process studied. That will be photosynthesis. That is, we have to define photosynthesis. Next, it may be asked that write the equation. Write the equation of reaction of process. Control, and so on. So the question is asked in this part. So what is the aim of the experiment? This is the aim of the experiment. Define the process, that is photosynthesis. Equation, you have to write the equation of photosynthesis, and then the control. So in this sequence, the diagram based question, experiment based question is asked. Now, the second experiment. Experiment number 2, to show that chlorophyll is essential. Chlorophyll is essential for photosynthesis. This time we'll select the plant which are not uniformly green. That is, here we take plant with a spotted leaf. Like croton, coleus, etc. Means these leaf of these plants are not uniformly green. Some part is green, some part is non-green, maybe yellow or some another color. Again, how is the experiment performed? First of all, we take the plant and keep it in darkness for 2 to 3 days in order to de-starch the leaves. Then the plant is kept again in sunlight for few hours. And then the leaf is plucked and tested for starch. But in that case, suppose we take the plant like this. Suppose we have this leaf. This is not uniformly green. Suppose this is green and this is non-green. So before going for the starch test, we make the outline of the leaf. We make outline of the leaf marking the green and non-green part. Green, non-green. This is before a starch test. Before a starch test. And then iodine is added, that is after a starch test. We'll observe that the part of the leaf which was initially green turned blue-black, and which was non-green turns brown. This shows that the green part of the leaf only performed photosynthesis and has a starch, having a starch. This shows that the chlorophyll is essential for photosynthesis. Now, this is the second experiment. Now, student, there is next factor, that is carbon dioxide. That is, carbon dioxide is essential for photosynthesis. CO2 is essential for photosynthesis. This time, again, the same step has to be taken. A well-watered potted plant, garden plant, is kept in darkness for 2 to 3 days in order to de-starch the leaves. And then we will arrange the setup. We will arrange the setup. How is the setup arranged, you see, students? Now we take a splitted cork and by placing over the leaf, the half part of the leaf is inserted in a conical flask in which some KOH solution is taken. The setup is then properly sealed, that is grease or oil is applied, grease or wax is applied to make it airtight. Now, the setup is then kept in sunlight. What is the purpose of KOH? So, KOH absorbs CO2 from the air inside the flask. Thus, the air inside the flask does not contain CO2. So, this part of the leaf does not receive carbon dioxide. Now, the setup is kept in sunlight for few hours, and then the leaf is plucked and tested for a starch. After a starch test, we see that the part of the leaf outside the flask which received atmospheric carbon dioxide performed photosynthesis. Hence, there is the presence of a starch and it turns blue-black, whereas this part turns brown. Now, this shows that CO2 is essential for photosynthesis. So, students, we have discussed 3 experiments related to photosynthesis. 3 experiments related to photosynthesis. Now, finally, the last experiment. Last experiment, experiment number 4, that is to show that oxygen gas is released. Oxygen gas is released during photosynthesis. During photosynthesis. Now, for this we have to take aquatic plant. That is, we will select the plant, some aquatic plants like it may be hydrilla. Hydrilla, it is aquatic plant. Hydrilla, Valisneria, Elodia, etc. So mostly we use hydrilla plant. While arranging this experimental setup, we take pond water in a beaker and this hydrilla plant is covered by short-stemmed funnel, which is kept over some support. Not its rim should not be directly touching the lower surface of the beaker. Short-stemmed. Then a test tube filled with water is inverted over it. Now you have to take care here that the level of water in the beaker should be above the stem or the opening tip of the funnel. Now this part. Then this setup is kept in sunlight. Gradually, when this setup is kept in sunlight, then we'll observe that some air bubbles start to rise, which gets collected in the upper part of the test tube. Gradually, the test tube is filled with this gas. The tube is taken out, and when glowing splinter, that is, the gas collected relights glowing splinter showing that it is oxygen. Now, thus we see that the oxygen gas is released during photosynthesis. Now, suppose, students, that if this setup is kept in darkness, the same setup is kept in darkness, then no air bubble will be formed and the tube will be completely filled with water. So, this will be the control setup. Suppose the question is asked that draw the control setup of this experiment. In that case, we'll just remove sunlight. The test tube will be completely filled with water. There will be no air bubble. That is, no oxygen gas is released since there is no photosynthesis. So this figure will be for the control of this experiment without sunlight. Now, one more question is asked. Suppose again, come to the next point again. Suppose it is kept in sunlight. Now the question is again asked. When a pinch of sodium bicarbonate or baking powder is added to water, then the rate of bubble formation increases. Rate of bubble formation increases. This is the answer. Question is asked that what happens when the pinch of sodium bicarbonate is added to water in the beaker. The rate of bubble formation increases. The reason is that this part, sodium bicarbonate, serves as the source of carbon dioxide, due to which the rate of photosynthesis increases. So, students, these were the different experiments on photosynthesis. Now, summarizing overall from all topics, all units related to photosynthesis, you have seen that stepwise we have explained it clearly, and I think you will be able to answer every question given in this chapter. But even if sometimes technically some question is asked, and if you will not be able to answer, then you can answer, directly ask the question by either writing in comment box or consulting us directly. Thank you.